Good afternoon to all. I am Dr. M. Job Gopinath, working as Assistant Professor in PG and Research Department of Zoology, Uris College, Vellore, Tamil Nadu. Today we are going to discuss about a very interesting topic called antibodies. In that, we explore the structure and function of antibodies. What do you mean by antibodies? Antibodies are the material which is generated by our body when antigens enter into our body. That is, antigen is nothing but a foreign material which may be a chemical, bacteria, virus and fungus. So these are called as antigen. When a body can have a antigen in it, automatically that will stimulate our immune system to produce antibodies. Antibodies are nothing but immunoglobulins. Why it is called as immunoglobulins? Because it is synthesized by the immune system and it is called made up of a globular protein. So that's why it is called as immunoglobulin. The structure of immunoglobulin shows it is in this slide. You can see the structure. It's a basic structure of immunoglobulin. It is Y in shape and it has two chains. One is heavy chain which is present in center and the other one is a light chain which is in peripheral region of the Y arm. This Y shaped structure of immunoglobulin is made up of different types of amino acid sequences. So based on that, we can see different varieties of regions, that is variable region and constant region in both light and heavy chain. It is made up of two chains, identical, that is light chain 2 and heavy chain 2, made up of amino acids with a weight of 50 kilodalton in heavy chain and 25 kilodalton in light chain. Each light chain bound to heavy chain by a sulfide bond at the base in arm region. The heavy chains are bound to heavy chain by hydrogen bonding. First 100 amino acids of amino acid terminal of Y region, that is in a fork region, you can see this is called amino acid terminal. In that region, both a heavy and light chains are variable region because this region has different varieties of amino acid sequences. But in others, it is called as constant region. We can refer as variable region light and variable region heavy. The same way, constant region heavy and constant region light. That antigen binding region is a complementarity determining region which can complement with the antigen. What type of antigen and what type of binding sites are there in the external shape of that antigen, based on that this immunoglobulin can be changed, the varieties. That's why this antibody binding site as variable region. It can vary based on the antigen binding sites or shapes which is present in the recognition sites in the plasma membrane of the antigens. Remaining center part is called as a constant. This Y-shaped antibody, again we can go to the structure. You can see there, it's here, there are different protein domains are there. In a typical Y-shaped immunoglobulin, two variable region in light and heavy chain, and in heavy chain three constant region, that is region or domains. It's nothing but domains, variable domain and constant domain. Whereas in light chain, one variable domain and one constant domain are present over there. So this is called, in Y-shaped light chain and heavy chain are there, it is called as an antigen binding site, whereas only heavy chain in the stalk region is called as a biological activity region. So antibodies are made up of proteins. So when we digest the protein with the enzymes, the bond between them are break down and automatically it gives a fragmented structure. In this slide you can see how a immunoglobulin molecule will be broken down into different fragments using pepsin, papain and mercaptoethanol reduction. We can see it later. In this slide we can see there are repeating domains of 110 amino acids are present in variable region. This region will be bind with intra-chain disulfide bonds with each domain. Heavy chain has one variable domain and three constant domain, that is CH1, CH2 and CH3. In some molecules CH4 also present. Light chain has one variable domain and one constant domain. Apart from this structure, it has a center part called a hinge region. This region has a proline residues based because rich in proline, it is very flexible in nature. This hinge region is found in only immunoglobulin G, immunoglobulin A and immunoglobulin D. Proline residues are targeted for the proteolytic digestion. That is, proteolytic means protein will be lysed by enzymes, that is papain and pepsin. And it is also, this region also rich in cysteine residues because of that disulfide bond. In IgM and IgE, these both immunoglobulins lack hinge region. Because of this lacking, they instead have extra constant region, that is called CH4 domain. In this picture you can see how each domain will be joined together by sulfide bonds in hinge region as well as in the heavy and light chain regions, or in heavy chains or in light chains. When we digest this immunoglobulin with papain enzyme, it yields three fragments, two identical FAB fragments and one FC fragment. What do you mean by FAB and FC? FAB means fragment that is antigen binding, and FC means fragment that is crystallized in cold storage. So this is called the FAB region, FAB domain and FC domains and everything, right? Now, when we digest this antibodies or immunoglobulin with pepsin, another enzyme, it gives only 2 FAB regions, and there is no FC region because this FC region is completely digested by the pepsin enzyme. In third, mercaptoethanol reduction, it eliminates the disulfide bonds and alkylation showed that. So till now we have discussed about what is immunoglobulin, what are the structure of immunoglobulin, what are the types of immunoglobulin, and how we can break immunoglobulin through this enzymatic digestion. In next class we can see about how we can produce antibodies in an animal through inducing the animal with antigen. It is very interesting. We can see it in next class. Thank you very much.